Welcome everyone. It's the afternoon in Australia, about 6 p.m. and morning in Europe. So it's the 30th April 2021. WebEVTalk EV Talk series for EV enthusiasts. So now today we've got a special speaker with a very exciting topic. Vincent Hyenne from INSERM Strasbourg in France. So Vincent, over the past 9 years or so, he has been using 3 complementary animal models, C. elegans, zebrafish, and mice, to decipher the mechanisms of EV secretion and their function in tumor progression and metastasis. So him and his team has notably identified the RAL GTPase as conserved regulators of exosome biogenesis acting on MVB's homeostasis. They also found that tumor EV secreted in a RAL-dependent manner accumulate in specific organs and promote metastasis, likely by inducing the formation of pre-metastatic niches. So today, his talk titled Biogenesis and Function of Tumor Extracellular Vesicles in Metastasis.
Okay.
Yeah, so please go ahead. Yeah.
Okay, thank you. Well, thank you very much, Jan and Carolina, for the nice invitation. And thank you as well for the initiative of this WebEVTalks, which have been very useful over this past complicated year. So today I'm going to present you our most recent work, focusing, as Carolina said, on the biogenesis of exosomes by tumor cells and how this biogenesis mechanism can contribute actually to the function of tumor EVs in metastasis. So this work is done in the tumor biomechanics lab in Strasbourg, France, which is led by Jackie Goetz. And so this is a plan of my presentation. So I'll start with a brief and non-exhaustive introduction on EV secretion mechanisms and what is known about the function of tumor EVs in metastasis. And then I'll drive you through our work of the past years. And it all starts actually with a study we did a couple of years ago now in a non-cancer model in the nematode C. elegans, where we identified a GTPase called RAL1 and found that it's involved in, it controls actually the secretion of exosomes in C. elegans. And then later on, we found that the autologs of these GTPase in mammals, which are called RAL-A and RAL-B, they are 2 genes, are also controlling exosome secretion in tumor cells. And not only that, but they control actually the function of the exosomes secreted by tumor cells. And they control a subpopulation of exosomes that seem to be extremely important to promote metastasis by reaching distant organs and forming these pre-metastatic niches. So here I want to emphasize that our work has been done in 3 different animal models. So C. elegans first, but now mouse and zebrafish. And I think it illustrates how important these animal models are if you want to understand some complex biological processes, and in particular, if you want to understand cell-cell communication in multicellular organisms. So our group is trying to understand the molecular and cellular mechanisms that are driving the metastatic cascade. So this is a process by which tumor cell will escape the primary tumor, reach the circulation, reach a distant organ, and invade it. So the group is split in 2 parts. One part is working on the biomechanical properties of tumor cells, and I'm not going to talk about it today. And the other part of the group, which I'm working on, is focusing on the extracellular vesicles that are secreted by tumor cells and how they can contribute to the formation of metastasis. And so we do so by working on mice and zebrafish mainly. So yeah, the audience is certainly aware of the high heterogeneity in extracellular vesicles. You have microvesicles, or 2 main types of vesicles, I'd say, microvesicles that bud from the plasma membrane, and exosomes, which come from an endosomal compartment, a multivesicular body, or MVB. And this endosome contains small vesicles that are called ILV, intraluminal vesicles. And upon fusion of the MVB with the plasma membrane, the small vesicles are liberated outside, and then they're called exosomes. So as you are certainly aware, There are many mechanisms now that are known of allowing the generation of these different types of vesicles. And we are focusing in the lab mostly on the mechanism driving exosome formation. So we are focusing on multivesicular bodies. And there, to form these intraluminal vesicles, you have two main types of mechanisms that have been described: escort-dependent and escort-independent mechanisms. And I just want to take a few minutes to describe one of these processes, because I'll use it later on in my presentation. It's a mechanism that has been described by the lab of Pascal Zimmermann in Marseille, France, at the center of which you have very special lipid, which is called phosphatidic acid, PA. And this lipid has some specific properties. It's negatively charged. It has a conic shape, and it has the capacity to induce membrane deformation. So the model proposed by Pascal Zimmermann and her group is that at the surface of the multivesicular body, you have an enzyme called PLD2 for phospholipase D2, which induce the formation of phosphatidic acid, so this lipid, on specific spots of the MVB, and that will drive ILV budding. And then this phosphatidic acid is bound to a complex of protein, syntenine, syndecan, and ALIX. And this whole machinery contributes to the formation of the ILV and, as a consequence, to the biogenesis of exosomes. Once the ILV are formed, the MVBs need to be transported to the plasma membrane, and this can be achieved by different RAB GTPases, the most famous one being RAB27, A and B. And then the MVBs will fuse the plasma membrane, and this is known to be controlled by a few snare proteins. So then these extracellular vesicles, which are very heterogeneous in content, also are secreted outside of the cell. And so, as you all know, they can contain lots of different proteins. Some of them are adhesion proteins, and they contain also lots of different types of RNAs, including coding and non-coding RNAs. And they will eventually be taken up by cells that are either close to the secreting cell or at distance. And in the case of tumor cells, this is extremely important, because tumor EVs were shown to be taken up both at the site of the primary tumor, but also they've been shown to be able to be transported by the blood circulation or the lymphatic circulation, and to end up in distant organs, where then they will be taken up by other cell types. So tumor EVs were shown to be functionally important for tumor progression and carrying mostly a pro-tumoral function. When they are taken up by other tumor cells, they can transfer some oncoproteins between tumor cells, but they can also transfer some specific phenotypes between tumor cells, such as the capacity to resist to some drugs or the capacity to migrate or to proliferate. When they're taken up by stromal cells, non-tumoral cells, they can induce phenotypic changes that have important consequences for tumor progression. They can be, for instance, taken up by endothelial cells, and that would promote, in general, angiogenesis and permeabilization of the vessels. And they can be taken up by fibroblasts or macrophages, and induce phenotypic changes that will, in general, lead to cells that are helping the tumor to grow and to form metastasis. Now, in terms of the real importance of extracellular vesicles in vivo, that the data we have now are coming from two main types of experiments, which are complementary. The first type of experiment are correlative experiments, in which people used tumor cells. I'm sorry, the light is going down because I'm not moving. Oh, I don't know. It's okay. I'll continue. Okay, so yeah. So in this experiment, people took tumor cells that were secreting more or less extracellular vesicles. They grafted them in the mice, and then they checked tumor growth and metastasis formation. And in general, when tumor cells secrete less of extracellular vesicles, they would form smaller primary tumor and less metastasis. And then you have another type of experiment that has been developed first by the lab of David Leiden, but then by other groups, which is called the priming experiments, in which you test more directly the function of the tumor extracellular vesicles. And here you grow tumor cells in culture in vitro, you isolate the extracellular vesicles, you inject them repeatedly in the mouse, And then subsequently, in the second step, you will inject tumor cells and check how metastasis is progressing. And so these two types of approaches allow people to come up with this model now where tumor extracellular vesicles are mostly pro-tumoral. They can modify the microenvironment around the primary tumor, but they can also travel in circulation and reach distant organ before the arrival of tumor cells, modify the microenvironment locally, and this is the formation of this so-called pre-metastatic niches. And then subsequently, the tumor cells will reach this organ, and because the microenvironment is favorable, they will form metastasis. So this is a very nice and exciting model, but of course, there's lots of questions that are not addressed yet. We don't really understand which secretion mechanisms are used by tumor cells to secrete all these different types of EVs. We don't understand how the heterogeneity of tumor EVs is important for tumor growth and metastasis. Then it's not clear how these EVs can reach the circulation, and also not clear how they can escape the circulation. There are some data reporting that tumor EVs do not accumulate randomly in organs, but rather specifically accumulate in the future metastatic organs. And this is the basis of this is also not very well understood. Also, the Leiden group has identified some adhesion proteins, integrins namely, that contribute to this process. Then what happens once the EVs are in this future metastatic organ is also not very clear. So this is the type of question that we are trying to address in the group. And now I'll take a step back and come back to a work we did a couple of years ago that brought us to what I'm now going to show you. So this is the work I did when I was a postdoc in the lab of Michel Labouesse in Strasbourg at IGBMC, where we were using the nematode C. elegans as a model system to identify new genes involved in exosome secretion. So this model is fantastic when you want to do genomic screens. And so we did an RNAi-based screen, tested over 1,000 genes, and trying to look for genes that would affect the cuticle of the worm, which is in this picture in the lower part of the screen. As the lab had shown before, that exosomes secreted by epidermal cells contribute to the formation of this cuticle. So we identified 73 genes involved in this process, and we decided to characterize one of them, which is a small GTPase called RAL. So RAL1 in C. elegans, and as all GTPases, it can fluctuate between two states, a GTP-bound active state and a GDP-bound inactive state. Its activity is controlled by different proteins, the most important one being RAS, which is a famous oncogene. Once active, RAL can recruit or activate a number of effectors, and these effectors will contribute to a large variety of cellular processes, exocytosis, cell migration, cell proliferation, autophagy, and others. And we decided to focus on RAL because of its known role in exocytosis of some types of vesicles. And we found out that RAL is controlling the biogenesis and the secretion of exosomes in C. elegans. And this is one of the phenotypes that we observed, which is in the RAL mutant, in the total absence of RAL in C. elegans, you have much less MVBs that are present in epidermal cells. And this was shown by electron microscopy, by a deep analysis. And these MVBs are actually bigger, and they contain less intraluminal vesicles. So these and other data that I'm not going to show today led us to this model where we think that RAL can act very early in the biogenesis or in the homeostasis of these multivesicular bodies. And because of this action, this will control the levels or the number of exosomes that are secreted by these cells. In addition, we think that in C. elegans, RAL could also act a bit later at a step of MVB anchoring or fusion with the plasma membrane. I'm not going to discuss that further, but maybe later on during the questions we can talk about this. So once I had finished this postdoc in C. elegans, I joined the group of Jacques Igutz in Strasbourg. As I said, he just had started his group on the metastatic cascade, and we thought it would be a good idea to continue working on RAL for different reasons. The first one is that RAL is conserved in evolution. It has two orthodox in mammals, RAL-A and RAL-B. And these two genes are known to be overexpressed or overactivated in multiple cancer samples and cell lines. And there are already several studies showing that in general, if you deplete RAL-A or RAL-B in tumor cells in mice, then you decrease tumor growth and metastasis. So therefore, we wondered whether the function of RAL in exosome secretion was conserved throughout evolution, and whether the role of RAL in tumor progression could be linked to its role in EV secretion. And this, with time, turned out to become the main PhD project of Shima Gorogi, with picture is presented here, and who defended a thesis last December. And so the work I'm going to show you is mainly her work. So to address this question, we used a cellular model. So it's a mouse mammary carcinoma cell line called 41, which is very aggressive and which mimics human triple negative breast cancer. So if you implant it in a syngeneic mouse model orthotopically, then you will form a primary tumor that will quickly metastasize to different organs, mostly the lungs. So we use these 41 cell lines where we depleted either RAL-A or RAL-B by shRNA, generating stable cell lines. And the first question we asked is whether when you don't have RAL-A or RAL-B, you affect the level of secreted extracellular vesicles. So we culture the cells in vitro, and then we isolated the EVs through different approaches, either ultracentrifugation or size exclusion chromatography. And the data I'm going to show you here are representing the analysis of the 100K pellet, so the small EVs isolated from these cells. And so what we found using electron microscopy, Western blot analysis of EV markers, and nanoparticle tracking analysis is that after depletion of either RAL-A or RAL-B, you have an important decrease in the number of extracellular vesicles, of small extracellular vesicles secreted by these cells. So in addition, we used two RAL inhibitors that have been described by the Teodorescu lab a couple of years ago, and which are called RBC8 and BQU57. And when we treat 41 cells or other type of tumor cells with this inhibitor, we also observe a decrease in the number of secreted extracellular vesicles. Also, this goes to different extents depending on the cell line. So it seems that indeed the function of RAL in extracellular vesicle secretion is conserved throughout evolution. To try to understand this phenotype, we first look at the localization of RAL-A and RAL-B. On the top left, this is the endogenous localization of the proteins, and you can see that there's strong colocalization with the MVB marker CD63. And on the right part, it's overexpression of GFP-RAL-A or GFP-RAL-B, and you can see also that it localizes in late endosome lysosomes, as shown by the colocalization with the lysotracker, but it also localizes at the plasma membrane. So then we looked at what happened to the endosomal compartments after depletion of RAL-A or RAL-B, and so we perform electron microscopy analysis of these cells through chemical fixation and high pressure freezing, so two different types of electron microscopy. And in both cases, we found that there is an important decrease of the number of MVBs present in these cells, as shown in this graph here. So this is the same phenotype that we observed after RAL depletion and C. elegans. So again, it shows that there is a conservation of RAL function, and it suggests that indeed RAL is important for the biogenesis of the exosomes. So to try to understand what happens downstream of RAL, we turn to this process that I described in the introduction, in which the enzyme PLD2 controls the formation of phosphatidic acid, and this drives the formation of intraluminal vesicles. So we decided to work on this because PLDs, so there are two, PLD1 and PLD2, are known targets of RAL in different processes independently of exosome secretion. So we wonder whether, in our case, PLD could explain RAL phenotypes. So we first look at the localization of PLD1 and PLD2 in 41 cells. And as shown on the left panel here, you can see that PLD1 very strongly colocalizes with RAL-A, and it's also the case with RAL-B, while PLD2 is mostly present at the plasma membrane and absent from endosomal compartments. Then we looked at what happens to PLD1 localization after RAL-A or RAL-B depletion, and here we found that this endosomal localization of PLD1 was lost in a large number of cells. So RAL seems to be important to properly localize PLD1 on multivesicular bodies. So then we wonder whether the function of PLD, which is to convert phosphatidylcholine into phosphatidic acid, was affected after RAL depletion. And so to address that, we performed some lipidomic experiments on the extracellular vesicles isolated from the cells, the control cells, or the cells depleted for RAL-A or for RAL-B, and we measured the phosphatidic acid over phosphatidylcholine ratio. And we found that after RAL-A or RAL-B depletion, there is a decrease in this ratio, suggesting that indeed, in the absence of RAL-A or RAL-B, the function of PLD is affected in these cells and in these extracellular vesicles. So then we try to inhibit PLDs, and for this we use chemical inhibitors against PLD1 and PLD2. And when we treated the 41 cells with this inhibitor, we also observed decrease in the number of extracellular vesicles secreted by the cells. And then if we use these inhibitors and we look at the endosomal compartments by electron microscopy in these cells, we also observed, so this is a graph on the right, we also observed a decrease in the number of MVBs present in these cells after treatment with PLD1 inhibitor, but not with PLD2 inhibitor. So these experiments show that inhibition of PLD1 is actually phenocopying depletion of RAL. And altogether, this data led us to this model where RAL-A and RAL-B would control the localization of PLD1 on multivesicular bodies, and that would be important for the homeostasis of these MVBs. And when you don't have RAL-A and RAL-B, you decrease the activity of PLD1, you have less phosphatidic acid, and you have, as a consequence, you have less multivesicular bodies in these cells, and then you have less exosomes that are secreted. So importantly, the cells that don't express RAL-A or RAL-B are still able to secrete extracellular vesicles, so in RAL-independent processes, and this could be exosomes or microvesicles. So then we wondered whether and how these vesicles could contribute to tumor progression and metastasis, and the first thing we did was to look in databases whether the expression level of RAL was associated with bad prognosis. And so we here on the top left panel, we looked at a large cohort of breast cancer patients from the TCGA, and this clearly shows that high level of either RAL-A or RAL-B is associated with poor survival probability in women with breast cancer. Then we looked a bit closer to this, looking at the expression of RAL-A and RAL-B by immunohistochemistry in the primary tumor of patients with breast cancer, with or without metastasis. And we found that there is a correlation between high expression of RAL-A or RAL-B and the presence of metastasis in this patient. So to try to understand this, we came back to our mouse model, and we used the capacity of the 41 cells to be injected orthotopically, so back in the breast lumen, in syngenic mice, in biopsy mice, and we measured over time tumor growth, and at the end point of the experiment, we measured lung metastasis. So regarding primary tumor, we found two different behaviors. The cells that don't have RAL-A, they grow faster and form bigger primary tumor, while the cells that don't have RAL-B, they form smaller primary tumor. But more interestingly, in both cases, we observed a very strong decrease in metastasis, and this was quantified on lung sections that are visible in this lower panel here. So even in the case of Raleigh, where the primary tumors are bigger, we observe a decrease in metastasis. So we checked different parameters of the cells, such as their capacity to proliferate or to migrate, and we didn't find any difference that could explain this decrease in metastasis. So then we turned back, of course, to trying to see if the EV is secreted by the cell at different functional properties. And for this, we perform the type of assays that I presented in the introduction, so the so-called priming experiment, where we isolated EVs from 41 cells expressing or not Raleigh and RALB, and we injected them twice in the mouse circulation, and then 24 hours later, we injected luciferase expressing 41 cells in the circulation, and we measured formation of metastasis in the lung over the next 14 days. And what we observed is presented here in this graph and in the pictures below. First, if you compare the brown to the gray curve, if you pre-inject 41 control EVs in mice, then you strongly increase the formation of metastasis in these mice. But if you inject the same number of EVs coming from cells depleted for Raleigh or for RALB, you lose completely this ability. So that shows that first, 41 tumor EVs are indeed pro-metastatic, that probably they modify the microenvironment of the lung and forming this pre-metastatic lesion. But EVs coming from SH-Raleigh or SH-RALB cells have lost this capacity. Yeah, so this is the conclusion so far, is that Raleigh and RALB dependent EVs promote metastasis. So then we tried to understand why, and we realized that if we wanted to understand what happens between the moment where tumor EVs are secreted and up to their uptake in a distant organ, it would be great to have a model where we could actually visualize circulating tumor EVs. And this is why we decided to use zebrafish. So before our study and the study from Guillaume Van Niel and Fred Verweij that we did in parallel, the main models used to visualize EVs in vivo were elegans, which is a great model. I love it, but it does not form real tumors. The Drosophila, which forms tumors but doesn't have a closed circulation system. And the mouse, in which you can visualize EVs up to a certain extent, but if you want to go to high magnification and resolution, then it's much more difficult, and you need relatively complex intravital setups. So this is why we decided to use zebrafish, which presents the main advantage, to be translucent, almost transparent when it's an embryo, and you can very easily perform fluorescent imaging, confocal imaging, and visualize objects with subcellular resolution. So as I said, we did studies that was published 2 years ago in parallel with Fred Verweij and Guillaume Van Niel that were studying the endogenous zebrafish EVs while we were studying the tumor EVs injected in zebrafish. So this is 48 hours post-fertilization zebrafish embryo. And what we did was to, in this study, we used zebrafish melanoma EVs that we isolated from culture cells, and we labeled them with a fluorescent dye that I'm going to describe in a minute. And we injected these EVs in a region that is connected to the heart of the fish. And once you do that, then the EVs will be quickly integrated into the circulation, and we image these EVs in the tail region of the fish, which is composed of a main dorsal aorta and then a complex venous system. So the goal of this study, and I'm just going to present it in a few slides, was really to describe what happens to circulating tumor EVs in vivo with high temporal and spatial resolution. So the dye we used in this study was called at the time membrite. it's a dye that is relatively bright, much brighter than the commercially available PKH dye, and that has the advantage to not form aggregates, fluorescent aggregates, by contrast to PKH. So we labeled EVs, and this is what you can see after injection very quickly. You can see lots of objects moving in the circulation, and some of them are very quickly arrested in minutes. They are arrested in different, taken up actually by different cells that we later on identified. Just to convince you that we are able to image small objects the size of an extracellular or small extracellular vesicles, we co-injected, this is the lower panel on the left, we co-injected 100 nanometer polystyrene beads, which are fluorescent in red, together with small extracellular vesicles isolated from these cells. And well, you can see that we are able to visualize both in vivo in the fish. So if the vesicles are sufficiently bright, we can certainly image them, even if they are below the resolution limit of our microscopes. So we characterize the behavior of these EVs in the circulation. So this is high-speed imaging of EVs in the dorsal aorta and in the caudal vein. And in the lower panel, you can see the red blood cells that are moving in the same vessel at the same time. And we found out that, by contrast to red blood cells, which remain in the center of the vessel, the EVs tend to spread all over the diameter of the vessel, and they come very close to the vessel wall, where then their velocity is extremely reduced, and we can see sometimes some EVs rolling along the endothelium and eventually get internalized by the endothelium. So the two main cell types that take up EVs, and this is what is shown in the lower panel here, are endothelial cells in red in this transgenic zebrafish, and macrophages in green in this fish. So after 10 to 15 minutes, most of the extracellular vesicles that we injected are arrested. And you can see in this movie, you can still see some EVs circulating in the venous vessels, and you can see others that are arrested, and they are already within endothelial cells or within macrophages that are extremely dynamic in this region. So just because I can't resist to show these movies, we characterize macrophages that took up EVs. These are very similar to the patrolling monocytes that exist in human and mice. So these are macrophages that are within the vessel, they are attached to the endothelium, and they send long protrusion to the lumen of the vessel. And through this protrusion, they can actually take up some extracellular vesicles. And this is what is shown in the movies on the right. You have two types of internalization of extracellular vesicles. In the first one, EVs will just stick to the surface of the macrophage. And after, in average 30 seconds, they will get internalized within the macrophage. While in the second type of uptake, the EVs are sticking to one of these protrusion, and very quickly, in a few seconds, they are moving to the center of the macrophage, and they get internalized at the basis of the protrusion. And so then we characterize the compartments in which these EVs will end up, and we found out that in macrophages, the EVs actually end up to late endosome lysosomes, and we showed that through a CLEM experiment. So CLEM stands for correlative light and electron microscopy. It's a technique that allows you to visualize an event in vivo in the living animals. So here, two macrophages that have taken up tumor EVs, and this was done in the living fish, and then we fixed the animal, and we found back those two cells by electron microscopy, and by looking at the 3D organization of the cells and of the labeled EVs, we were able to show that EVs accumulate indeed in late endosome lysosomes. Okay, so overall, we've shown that this model could be very useful and very complementary to study the function of tumor EVs in vivo. And we used it in what I'm going to show next. So coming back to the RAL story, we knew that RAL-A and RAL-B dependent EVs are promoting metastasis, but we did not really understand how that could work. So the first thing we did was to label EVs using this membrane dye and inject the labeled EV in the circulation of the mouse. And after 1 hour, we sacrificed the mice and looked at different organs, and we observed strong accumulation of 41 EVs in lungs as well as in liver, as shown here. But when we injected the exact same number of labeled EVs coming from SHRA or SHRB cells, we observed much less accumulation in the lungs. And this is confirmed actually in the right panel with this lung section, where you can see that there is also, we observe a decrease of EVs accumulating in the lungs when these EVs are coming from SHRA or SHRB cells. So to determine whether these EVs had limited capacity to be taken up by endothelial cells, we tested this first in vitro on the left panel on culture UVEC cells, where indeed the same number of EVs coming from SHRA or SHRB have reduced capacity to be internalized by endothelial cells. And similarly, in the in vivo zebrafish model, we could observe a slight decrease in the accumulation of EVs coming from SHRA or SHRB cell. So these EVs seem to have defects in adhesion or internalization properties. To try to understand why, we looked at the content of EV secreted by control and SHRA and SHRB cells. We observed important differences in the RNA content, but I'm not going to describe that further unless you have some question later on. But then we looked at the proteins that are expressed by these EVs, and we found a number of them that are differentially expressed between EVs coming from control cells or EVs coming from SHRA or SHRB cells. And 5 of these proteins, which are listed here, are systematically overexpressed in EVs coming from control cells versus EV coming from either SHRA or SHRB cells. And some of these proteins were validated by Western blot. And in this Western blot, of course, we load the exact same number of EVs. So we decided to focus on one of these proteins, which is CD146, because it's an adhesion protein. It has 5 immunoglobulin domains. It's also known as MCAM, and it's known to be extremely important for adhesion of different cell types to the endothelium. And it's also involved in metastasis in some mouse models. So we wondered whether CD146 MCAM could actually be responsible for the adhesive capacity of RAL-dependent tumor EVs. And to test that, we use an antibody, a blocking antibody directed against CD146. And we first observed that when we treat 41 control EVs with this antibody, we decrease the uptake of EVs by endothelial cells in vivo in the zebrafish embryo. More strikingly, when we treat the 41 EVs with this antibody and when we injected this in the mouse circulation, we also observe a decrease in the accumulation of these EVs in the lung. And finally, we perform this priming assay where mice were pre-treated either with EVs treated with a control antibody or with EV treated with an anti-CD146 antibody. And later on, luciferase 41 expressing cells were injected in the circulation, and we measured metastasis. And again, if the mice are pre-treated with control EVs treated with a control antibody, you have an increase in metastasis. But if the mice are treated with EVs incubated with blocking anti-CD146 antibody, then you have a decrease in the metastasis. So, which is a phenotype, of course, similar to what we observed with EVs coming from SHRA or SHRB cells. And finally, we checked in the TCGA databases, and we found that in the same cohort of breast cancer patient, which is a large cohort, high expression of CD146 is associated with a decrease in survival probability in women with breast cancer. So finally, we propose this model where highly metastatic cells in primary tumors secrete large number of extracellular vesicles, part of them through RAL-A and RAL-B PLD1 PA axis that governs the MVB biogenesis or homeostasis. And the EVs that are generated in a RAL-dependent manner, they are enriched in some specific proteins, including CD146. And because of this, and because of these proteins, they have the capacity to accumulate in specific organs, mostly in the lung, where they will change the microenvironment locally, and this will promote the formation of the pre-metastatic niches, and this will then help tumor cells that will arrive in second time to grow more aggressively. And so this model is supported by a very recent data that we obtained and which are not published, where in collaboration with Etienne Coyot and Isabelle Fournier in Lille, we analyzed the proximal interactomes of RAL-A and RAL-B using the BioID approach. And we identified a number of proteins that are interacting with RAL-A or RAL-B, but importantly, two of them are very important for the story I just told you, as we identified PLD1 in the RAL interactome, but not PLD2. And we also identified CD146 as one of the top hits of proteins interacting with RAL. So in the absence of RAL, tumor cells are still able to secrete extracellular vesicles, which also to a decreased number, sorry. But importantly, these vesicles have much reduced capacities to promote metastasis as they accumulate less to the lungs and they are less efficient in promoting metastasis. So this also illustrates the importance of trying to understand the heterogeneity of extracellular vesicles secreted by tumor cells, because this could allow us to identify precisely which subpopulations are important to target eventually with an optic of therapeutics. Okay, so with this, I'll finish. I just want to thank all the collaborators that help us to gather all this data and to perform this project, all the funding agencies, and most importantly, the people that directly did the job. So as I said, the lab is led by Jackie Goetz, and many people of the lab contributed to this work, but most importantly, Shima Gorogi, former PhD student now, and Benjamin Marie was currently in PhD, are the two most important person who worked on this. Okay, so that's it. I'm stopping here, and I would be happy to take any questions.
Thank you very much. How do you pronounce your name? Vincent?
Vincent, yes.
There you go. Thank you very much for a very interesting presentation and some very, very interesting stories. Before we go to questions, please turn on your video and allow Carolina to take a picture of us all attending, or some of us at least. So that'd be great. And smile, right? Yes.
Behind the mask.
Wear a mask if you need to. Absolutely.
Okay,
are you done, Carolina?
Everybody smile. Not yet, not yet. That was,
okay.
We need to take 3 times actually, because we have quite an attendance today. Okay, keep turning it on if you haven't done so. Okay, another one.
Yeah, we have 3 screens full. It's good.
Yeah, all right, all good. Go ahead.
So I'll let Anaï Grozzo unmute and ask her question, please.
Sorry, I just turn on the
Hi,
setting.
yeah, can you hear me? Yeah.
Yes.
Very interesting talk. Thank you very much. Do you think that EV's extravasation would be more efficient if they are taken up by circulating monocytes first?
I don't know. That's the answer. We never tested that. We do see sometimes that some of the patrolling monocytes that have taken up EVs are found behind the endothelial barrier. Okay, so it's a possibility, but I have no idea whether then they can be secreted again. We have absolutely no evidence for that. So the only mechanism that has been described so far is, or the 2 mechanisms, is through endothelial cells, either because the endothelial cells are permeable or through transcytosis through the endothelium.
Okay, thank you.
Any more questions? We're not going to finish already, are we? Keep on asking. Christina Liboni, please unmute and ask.
Hello, everybody. Thank you at the beginning for the very nice talk, very amazing work. I was wondering how AVs could trigger the formation of the premetastatic niche. I mean, when they arrive, do you have an idea which kind of actions they can exert on the site of future metastasis? Thank you.
Yes, so we did not explore this yet. I mean, this is something we want to do, but it's known from the work of several groups that there are many events that happen once the tumor AVs arrive on the premetastatic niche. The first one is creation of an inflammatory environment. The second one is modulation of the extracellular matrix. And then you have the recruitment of different type of immune cells to this site, and probably secretion of specific growth factor as well that will modify the microenvironment. So you can look at the work of David Leiden, for instance.
Can I follow up on? Is there any chance at all that EVs could carry oncogenes and transfect cells at a different site, creating a new clone of tumors? You think that happens?
Well, I mean, this has been shown in vitro, right?
Yeah. But in vivo,
but never in vivo, to my knowledge.
No, I haven't seen anything.
In our case, in the content analysis we did, we didn't find oncogene with specific oncogenic mutations. So, yeah.
So Roberta Palmuli, please unmute and ask a question.
Yes, I was wondering more about the cell biology and the trafficking of the RAL protein. So do you know, for instance, how RAL1 is recruited to what seemed to be a subpopulation of MVBs if you have other RAS protein or GTPase, and if you know the cascade? And also, I mean, you find PLD1, but not PLD2, and both of them seem to produce PA. So, I mean, why one or not the other, how this could happen? And also because you find then very specific cargos again. Yeah, if you have like some clue of why this specific cargo may finish in PARH ELVs or.
Okay, yeah, thank you, Roberta. So the first question is what's acting upstream of RAL in their MVB localization? So we don't know, but this is something that we are now studying. We are mostly trying to see whether RAS activation can promote RAL activation on MVB surface. But yeah, I don't have the answer yet. So then regarding the PLD issue, it's true that Pascal Zimmerman lab found that PLD2 is involved in exosome secretion. And they did their study in a breast cancer cell line, actually MCF7. Yeah. So you could think that it's relatively close to what we are looking at, but we don't see PLD2, but rather PLD1 involved in our process. So the reason, I mean, the evidences we have for that is that in 41 cells, PLD2 is at the plasma membrane, while PLD1 is on MVBs. And then when we inhibit PLD1, we observe a decrease in the number of MVBs in the cells. But when we inhibit PLD2, we don't see this phenotype. Okay, and finally, we have this interactome data showing that RAL interacts with PLD1 and not PLD2. So clearly in our case, I'm convinced that it's PLD1. In their case, it's quite convincing that it's PLD2. So the only explanation so far that we can have is that different cell types, different mechanisms. And I mean, it's been shown for other exosome secretion machinery, for instance, work from Clotilde's lab showing that RAB27 is important for 41 exosome secretion and tumor progression and metastasis, but not in another mammary tumor cell types, which is called TSA. So clearly, depending on the the cell type, different types of machinery are used to secrete extracellular vesicles.
So Esther Nolte, please unmute and ask.
Yeah, thanks, Vincent, for this great overview of data. It was very nice. I was actually wondering, would you think that the interference in REL-A, REL-B could be a more general strategy to distinguish between exosomes and microvesicles, at least maybe in tumor cells? In relation to that, and more specifically, what exactly did you see in the EM? Was there a general decrease in intraluminal vesicles in all multivesicular bodies, or were some gone and other multivesicular bodies were still full of intraluminal vesicles?
Yeah, so regarding your first question, no, I don't think so. Because actually both RAL-A and RAL-B are also present at the plasma membrane. And we first focused on MVBs and exosomes because of the work we did in C. elegans before. But now we also looked at the 10K pellet, which is supposed to have bigger vesicles, eventually more coming from the plasma membrane. And here, interestingly, when we deplete RAL-A, we see a decrease, but not when we deplete RAL-B. So we're not fully understanding this yet, but this could mean that RAL-A is maybe involved also in the secretion of microvesicles, but not RAL-B. That's one thing. So then regarding the MVB number, so the main phenotype we observe in 41 cells is a decrease in MVB number, not in ILV number. Okay, but this is after depletion of either RAL-A or RAL-B. So now we generated cells double knockdown for RAL-A and RAL-B. So then we observed a little bigger decrease in the number of EVs. We have a little bit less MVBs. And here in these MVBs, we observe less intraluminal vesicles. Okay. So now, whether it's all the MVBs that have this phenotype or not, I'm not sure. I mean, this is an average decrease, right? But some have more than others. But it's hard to tell from our electron microscopy approaches, because what we do is to take single EM slices randomly, and we accumulate a large number of MVBs. But and we can do stats on that. But when you look at one single MVB, you don't know where you are in the MVB. You could be very well at the top of the MVB or at the center of the MVB. So it's hard to conclude from one single section. If you want really to characterize this, then we'll need to have a serial section and 3D reconstruction of MVBs to see how different they are. And this is something that we are also planning to do.
There are multiple variables. One is volume or three-dimensional localization. The other one is time as well. Things can happen quite quickly in cells. Esther, I interrupted you. Did you have follow-up?
Yeah, I'm fine.
Esther?
Yeah, no, that's fine, Jan. I was just wondering, I can't remember whether you showed, but so there is not a complete co-localization of RAL-A and B in general?
Yeah, there is. Yeah. So
Okay.
these two proteins are known to work together in lots of different contexts. Yeah.
So Aaron,
Thanks.
please unmute yourself and ask a question.
Hey Vincent, thank you. That was a really interesting talk. I'm just wondering, as a quick question really, whether you ever see your labeled EVs being taken up by cells other than endothelial cells and intravascular macrophages.
By other cell types, you mean?
Yeah.
Yeah, so we don't see, so we tried neutrophils and we didn't see any internalization. But then when we injected in the EVs in the GATA1 cell line, what label red blood cells, but also hematopoietic stem cells that are in the caudal vein, then we see some of these cells that are not motile that accumulate EVs. So presumably some hematopoietic stem cells also take up EVs there.
Is that in the CHT as well? In the
yeah.
Yeah.
Yes, exactly. Yeah.
I have a question which is more philosophical and strategic and even political, maybe. I think your work is amazing and it's really important from a cell biology perspective and basic research perspective. A lot of the funding that comes to biological research goes through medical funding agencies. NIH is huge. Everything has to be connected to a disease somehow. do you feel burdened, or you're limited by having to include cancer kind of associated research in your cell biology? Because, you know, I think it's good research. There's nothing wrong with it. But if you just, wouldn't it be nicer just to be able to focus on the mechanisms and not bother about diseases at these stages?
Yeah, I used to think like that at some point, especially when I was working on C. elegans.
Yeah.
Now I'm really in a cancer environment, and I see other interest in that. And clearly, I mean, this question of the pre-metastatic niche is very unique to this disease, and I think is fundamentally super exciting, actually. So I see advantages, actually, to study cell biology of EVs in a cancer context that are very specific to cancer questions. And then eventually it could also help. And I mean, I'm not against that.
No, I mean, also basic research, blue sky research can result in therapeutics or diagnostics or things like that. But okay, thanks for your response. Gwenan, please unmute yourself and ask your question.
Yeah, thank you. Thank you, Vincent. It was very cool to see all your stories together. It was really clear. I was just wondering, I forget, or maybe I miss it. Do you have any effect on the viability of your cells if you block either PLD1 or rare?
Yeah, there's a very, very slight decrease in the mortality of the cells, but it's really at the margin. Yeah.
Okay. Thank you.
Any more questions?
Yeah, may I ask a question? Stéphane Brésillion from Reims.
Okay, well, you should put the question in the box, but you can, Stéphane. Okay, go ahead.
I noticed that GPC4 was downregulated, and it means that glypican-4, I guess, is downregulated. Vincent, can you tell us a bit more about it? Is it just downregulation of the protein expression? Do you see a change in the GAG chains, in glycosaminoglycan chains? Do you see a change in the smear?
No, that I don't know. We just found it by mass spec, and then we confirmed it by Lotvall. But no, I cannot comment on that.
Okay, thanks.
Sorry.
Okay, before Aaron comes back, I'll ask Louise Hubert to unmute and ask a question, please. Wait, you're
can't hear you.
You have to unmute.
Yes, sorry. Hi, I'm a French master degree student, so maybe my question will not be very relevant, but you have shown that with a knockout of RAL A and B, the metastases are less present. But can we imagine that the knockout of CD146 could therefore prevent the binding and therefore the microenvironment will not modify and will not allow the formation of metastases?
Yes, exactly. This is exactly the idea we have, is that CD146 will be required for the accumulation of tumor EVs in the distant organs and so the apparition of the pre-metastatic niche. We just generated 41 cells depleted for SH146 and will now test whether indeed these cells have reduced capacity to form metastasis in mice.
Okay, thank you.
Okay, Aaron, please unmute and ask a second question.
Okay, so it's just, it kind of comes back to the first question, really, and just wondering about how exactly, so I guess both sides of the situation, so how the EVs from the tumor cells got past the endothelium to get into the circulation, and then the other end, how they're able to get out to prime that pre-metastatic niche. And I was just wondering about how you might approach that, and obviously it'd be really neat if you could do it in the zebrafish to visualize and then start to dissect the mechanisms that are involved. But am I right in thinking that it's quite difficult to model metastasis in zebrafish? And I'm just wondering if you've got any ideas on how you could begin to approach that.
Yeah, so for the functional question, we did some functional assays similar to the priming experiment performed in mice, but in zebrafish then. So we injected the EVs at 36 hours and then at 48 injected the tumor cell in the circulation, and we observed an increase in the metastasis. So this was in a setup using zebrafish melanoma cells and EVs. Okay, so it's possible to do functional assays in the fish, and this is what we are pursuing now. So then the question is how to image the tumor EVs escaping primary tumor and reaching circulation. So that we never tried. The opposite, we are trying to look at it, but we never observed EVs, or very rarely, behind the endothelial barrier, which is a bit different from what Fred observed, for instance, where he observed some of the YSL EVs going behind the endothelial barrier. But again, in our case, you have to keep in mind that we used fluorescently labeled EVs with a membrane dye. And after a certain time, you have still always to wonder what you are looking at. Is it still the vesicle, or is it just the dye? Yeah. So we are also generating fusion proteins to try to see if there is any difference.
So similar to Frederick's approach.
Yeah, well, different proteins, but similar idea.
Fusion reporter, yeah. Brilliant. Yeah. Nice one. Thanks again.
Thank you.
Any more questions?
One quick question. Vincent, so have you injected the EV longer than 36 hours? It seems to be very short, and whether that's if you done so, you see any difference or not?
So you're talking about the priming experiments, right? Yeah. So we did a few tests with longer and more frequent injections, but we lost quite a lot of mice. Oh. So at the time we were isolating EVs only by ultracentrifugation, and some of the mice were dying. So now we're using side exclusion chromatography. We don't have this anymore, but we switched to this protocol because it was working. But I think that's what's really lacking in this field. I mean, many labs are doing this type of priming experiment, but all the labs have different protocols. So there's no consensus of what should be done on. And I think we should
Why did you lose the mice?
Well, they died very quickly after the injection, so...
Disseminated intravascular coagulation or something?
Yeah, I guess.
Thrombosis.
So. Yeah, I think that's...
Tissue factor on EVs or something?
Yeah, or aggregation. I'm not sure.
But tissue factor, was that from platelet EVs? Do cancer EV also has the same? Not sure.
I'm not sure.
But
we got rid of this problem when we use with sec isolation of EVs. So
you're using SEC at the end?
For the in vivo experiments, yes.
Okay.
If no more questions, thank you very much, everyone, for participating. This was a really enjoyable session. We see each other again in 2 weeks, and what the topic will be is going to be presented by Carolina, and you also talk a little bit more about other activities.
Yeah,
thank you very much.
Thanks so much, Jan, for leading the QA. It's wonderful as well. Vincent, thank you so much for the presentation.
Thank you very much again for the invitation.
Yeah, very exciting data and also lovely images. I really enjoyed the whole talk. Thanks so much.